Now that we've learned about the phospholipid bilayer, we're ready to talk about transport across the cell membrane. And the first piece of that is going to be passive transport. That should give you a hint. We're going to get to active transport after that. So what this relies on is a gradient. Things are going to diffuse across the membrane down their concentration gradients. We're going to get to electrical gradients as well. So remember, chapter 1 talked about how gradients are so important in physiology, thermal, pressure. This is going to be primarily concentration gradient. So the amount of a chemical, it's also called a chemical gradient. So you know about diffusion already and gradients. If you think about freshly baked cookies, or it could be a good smell like that, could be a bad smell that comes from somewhere in the classroom or your dorm. Those odor molecules are chemicals that are in high concentration wherever the source of that odor is. As those odor molecules spread or diffuse, they go from high to low concentration, molecules randomly moving about such that they evenly spread out. The net movement of the molecules is to spread. So particles tend to distribute themselves evenly within a space. This is air in this example. The same is true for a solution, some sort of liquid. And again, in our bodies, that's going to be water. So movement's always from high to low concentration, down the concentration gradient. And your book talks about that as downhill. So this same idea that's shown here would be true for a container, right? If you had a container of water, I don't want that one, I want this one. And if you had a small area with a bunch of molecules in a container, over time, those would spread to be evenly distributed throughout the entire space. The same idea is true for membranes. Materials move across a membrane by diffusion as well, from high to low concentration. And then the important thing about this is spontaneous. So it's passive transport, and then equilibrium is reached once you've got this even spread. There's still some kind of random movement, but there's no net movement. So we're going to go back to the membrane in a moment. First, what I want to do is talk about the factors that influence the rate at which this happens, this diffusion, because this is important for diffusion across the membrane. So factors that influence diffusion rate. And I actually have 5. So look at this. I can just do this right here. I can do 5. I also want to erase this stuff. Oh, there we go. Awesome. Okay. So we've already talked about molecule size. That's one reason some people classify these differently, right? The larger the molecule, the less easily it's going to pass across that phospholipid bilayer. So water can move a little bit better than glucose, and glucose is hydrophilic, but also is a little bit large. So other things that matter are going to be temperature. So this is the kinetic energy in the system. The higher the temperature, the faster molecules move around, and the faster equilibrium will be reached. Molecule size, that's the one I just said. Concentration gradient, so the size of the difference across the membrane. We'll see some examples of very big gradients in this in the future. Distance, so the thickness of the membrane. This doesn't vary for the plasma membrane because the plasma membrane always is about the same thickness, because the plasma membrane always looks like this, might have proteins in there too. Ooh, I like this tool. But this is going to matter when we start talking about diffusion across body membranes. So for example, for respiration, that oxygen and carbon dioxide has to pass across multiple plasma membranes as well as basement membranes that are made up of tissue. So we're gonna, and then how many cells it has to go through also matters. So distance will be important, not important for looking at one plasma membrane versus the other. And last is charge. This is about the electrical charge, and this will become important as well. I'm not going to talk about it now. So now I've got that ugly stuff there. The idea here is that when you start with these two, now we have two different compartments. One side has red molecules, other side has silver. Over time, if the membrane is permeable, which is what these dashed lines indicate, that indicates permeability or semi-permeability. It's permeable to something. In this case, it's clear it's permeable to these molecules because the red's moving this way, the silver's moving this way to reach equilibrium. So this is what this looks like across a membrane. We've got more molecules on this side at the start. This is time going across this way. And this might be glucose. Our lipid bilayer is going to let this molecule in enough in this case. So right now, we're not really talking about whether something is permeable. We're talking about gradients. Two things that matter for a molecule to be able to pass across a membrane is it able to pass across physically the phospholipid bilayer or proteins that may be present? And two is there a drive for it, a gradient, or do we need energy to do it? So right now we're talking about the energy input, which is diffusion is the passive piece. So over time, these molecules start to diffuse. They move across the membrane down their concentration gradient. Over time, the amount of time depends on the size of the molecule, if this was a different thickness, the temperature, the charge, all those other things. So now let's go back to, if we want to have something pass across here, not only does it have to have a gradient, but it needs to be able to go across the phospholipid bilayer, or we need a protein in there that will help us get it across. Let's go back to a learning check. I want you to pause the video and answer this question about constructing a molecule that could pass through the bilayer. How do you want to make this molecule be? Okay, it should be small and hydrophobic. Small so that it can pass through physically, and hydrophobic because the core of that membrane is hydrophobic, and a hydrophilic molecule is not going to spontaneously pass through. Just not going to happen. It's chemistry. Example of this would be oxygen and carbon dioxide. So here's that chart I showed you in the last lecture that you could review here if you need to in terms of what can cross across the phospholipid bilayer and what cannot. So the bilayer is selectively permeable. So now we're going to go back to, if we want to be able to get a large amount or any amount of these ions into the cell, or even glucose, what do we need? We need a membrane protein. So that's going to change the permeability of our membrane, and that's good. I mean, it's important. We need to be able to get things in some cells when we want to, and etc. Again, membrane permeability means allowing things to go through. High permeability means it allows that to go through. So here's a quick review of the membrane proteins I talked about in one of the previous lectures. And I told you then, it's still true. The focus is going to be on channels and these two types of channels, as well as carrier proteins. These are what are the transport proteins that allow things to pass through. These other types I'm not going to go over again right now, but these are other membrane proteins that are important. So, passive transport across the membrane, also called diffusion. That's what we're talking about, right? We're talking about something that has a gradient, has a drive to go across. In this case over here, what might this be? Well, lipid soluble, tells you right there. This could be oxygen, O2, or CO2, because this thing can go right across the plasma membrane. It's small and hydrophobic. Simple diffusion is what this is called. simple diffusion. If we want to get something across that can't go across the hydrophobic core of the plasma membrane, the phospholipid bilayer, we need a protein to help us. So these two types here are both facilitated diffusion. Facilitated means helped, right? If you facilitate someone, you help them. These are facilitated diffusion, still types of diffusion, meaning it's still down the concentration gradient. The two types here are carriers. That's here, carrier molecules. We'll see examples of these. Typically amino acids and glucose are the two that are going to use carrier molecules. And then these are channels. These are going to be typically for smaller molecules. So ions, small lipid insoluble. So again, these are very similar, but the idea here is this is often small. We're going to see ones for potassium, sodium, chloride, calcium, say that already. So smaller typically. And then importantly for these channels, there's going to be gated channels and non-gated. So some that are always open and some that need a stimulus to open. Non-gated are also called leak channels. So we'll come back to lots of examples of these. One more example here. This osmosis is actually a special type of facilitated diffusion. It is diffusion of water. And so this aquaporin facilitates water movement. Some water can cross across the membrane, the phospholipid bilayer, but aquaporin is a channel, a water channel. It's non-gated. So that's a good example of a leak channel for water. And we'll talk about osmosis in a future lecture and in lab. Quick learning check. Why does glucose need a carrier protein? Here is a glucose molecule. This is the carrier. It needs it to get through. Why? Here are some choices. The answer is B. There is a concentration gradient in this situation here. There wouldn't always be, but in this situation there is. However, glucose cannot cross the phospholipid bilayer. So this facilitated diffusion is what is shown here. That is when something cannot cross the phospholipid bilayer on its own, it needs a protein to be able to pass through. Something about these proteins that might help this tie together a little bit more is, what do you think the chemical properties of this protein would be like right here? It would be hydrophobic, so that it can be stable in that membrane there. This portion of the protein would be hydrophilic. So these proteins are specialized to allow them to be transmembrane proteins. And then something about this protein also allows it to bind to glucose. So protein structure is very important here.